This meeting is in continuation of our previous meeting and the previous YouTube video. Okay. So if I share my screen, can you see? Can you see my screen?
Yes, sir.
Can you see these displayed?
Yes, sir.
Okay, this is your blue-white selection. So you see how blue colonies are and how white colonies are. So our interest is in white colonies because these are the colonies that will have our gene of interest. And blue colonies are empty vector religated, so they will not have a gene of interest. Okay.
Yes, sir.
So and another property is that it should have high copy number. So this lecture is in continuation of the cloning lecture, and here we are studying the properties of your cloning vector. So here another property that we talk about is high copy number. That means one E. coli cell will harbor a lot of plasmids. That means not one plasmid but many plasmids. So two different types of plasmids cannot stay inside one cell because of the process we call as plasmid incompatibility. Okay, but similar kind of plasmids can stay in a cell. Okay, and like for example, if we there's a vector called as PUC18. Okay, we call this vector, this is plasmid, this P is small, C is capital, is the University of California developed this plasmid number 18. So it has 500 copies in one cell. Okay. So we prefer that our host in which we clone or a vector should have the property that it should have high copy number, because if you have one copy number, if at the end of a cycle you have say 1 million cells, so you'll have 1 million plasmids. But if you have a copy number of 500, that means each cell will have 500 plasmids. That means 1 million into 500 is around 500 million plasmids at the end of your experiment. So you'll always prefer that. So why this happens is we make the host, okay, faulty in replication. But when replication occurs, okay, rolling circle replication or whatever, D-loop replication. So for example, it's a rolling circle replication. This is a plasmid, okay. There's a nick, okay, and now the condition is like that. This is not your syllabus, but it's not bad if you do not know this. So now replication occurs here. Okay, it goes like this, and this strand is peeled off like banana. Okay, this again moves. This is again peeled off until you have the whole, and this one recircularizes. Sorry, this one recircularizes, and second strand is synthesized. So this one is now two strands. What will happen? This will stop replication here. Okay, because the process that there are proteins called as riproms. Okay, they bind to the site so that replication does not refire. We call it as refiring of the origin of replication. So if we mutate those riprom proteins, okay, that bind here, it will recognize it. It will not recognize that it has. We have to stop here. So what will happen? It will go again, keep on going. So this will peel off. This will again keep on going. So this will peel off. And at the end, again, you have two plasmids, and this will peel off and make another plasmid. So by refiring of the origin of replication, you have multiple copies, like mini copies, like 500 copies in your bacterial cell. So these are the properties of some of the properties of your cloning vector. Now, next step is your preparation of the insert that we studied. So you have an insert either, see, if you want to clone genomic DNA, it's simple. If you want to clone a gene, it's a little bit complex if it comes to eukaryotes, because in case of eukaryotes, in case of, say, for example, in case of prokaryotes, you take the whole genomic DNA, okay, you take the vector, digest it with EcoR1, digest this with EcoR1. Okay, so you have so many inserts. Insert 1, 2, 3, 4, 5, 6, 7, and many. EcoR1 will digest, okay, 4 to the power 6 times. The probability of digestion of EcoR1 or recognition of EcoR1, okay, if it recognizes here, it will recognize after 4 to the power 6 bases. But we do a partial digestion, like for a smaller time, or we use less enzyme so that we get larger fragments of the genomic DNA. So what we do, we got now these fragments 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then you have vector. So since it's a mixture of vector, you have some millions of copies of vector, okay. You again digest this with EcoR1. So you have like this. Okay, now you mix these two. Okay, you take this and you take the vector. We take the insert, this is the gene or the DNA we are going to incorporate, we call it an insert. Insert to vector ratio 2:1. Insert we take more because the probability is that the vector will ligate self-anneal. So, but if the inserts are many, the probability is that it will religate with the vector. So you digest genomic DNA. So this is how we make a genomic DNA library. It's sort of cloning the whole genomic DNA of a bacterial cell, say for example, or plant cell. Say in case of, we'll first take bacterial cell. So we took the DNA, digest it with EcoR1, take the vector, digest it with over EcoR1, mix the two, okay, in the ratio 2:1. Then we add ligase. Restriction enzyme does not require ATP, okay, because this breaking of bond, it releases energy, but ligation, we require ATP. So the buffer that comes for ligase consists of ATP, okay. So we mix the vector and the simple mixture, take 2 microliters of insert, 1 microliter of vector, 1 microliter of ligase, and say 2 microliter of buffer, then add water. And also you should keep the volume of the reaction very low, because higher the volume, more the dilution. So vector and insert will be parts away. But if you keep the volume low, say 10 microliter reaction, it will be good. So you have a vector, 1 microliter, you have insert 2 microliters, you have ligase 1 microliter, you have buffer say 1 microliter, and you have water 1, 2, 3, 4, 5 microliters. So total volume is 10 microliter reaction. Then what you do, the ambient activity of a restriction enzyme is 37 degrees centigrade. So when you restriction digest, you put it in a water bath at 37 degrees centigrade. Ambient activity of ligase is also 37 degrees centigrade, but we do not keep it in at 37, because at 37 the kinetic energy of the molecules is high. For example, so that's what we do. When we do this, we keep ligation at 16 degrees centigrade or 8 degrees centigrade. So simply we take the tube, mix everything, and then put it in the fridge in the lower corner. So the refrigerator that comes, the lower shelf where the vegetables, we keep vegetables, it has a 16 degree temperature. You go up, it has 8, then it has 4, okay, and the freezer, it has sub-zero. Now, for example, this is your vector end, and this is, and why do we use 16 degrees centigrade? Okay, we do not use 37 degree for ligation. We use lower temperature, 8 to 16 degree temperature. It is not the ambient activity of ligases, but still it will have some activity. Okay. So now this is the insert. Okay, so this is the insert, and this is one end of the vector. Okay, now they have to ligate, for example. Okay, Now we do the ligation at 37 degrees centigrade. At 37 degrees centigrade, the kinetic energy of this molecule is high. Okay, so there is random motion. So now I want to ligate it. Can I be able to ligate? No. Okay. Now we lower the temperature 16 degrees. So kinetic energy decreases. Okay. Now if I try to ligate them, they'll be able to ligate. Right. So this is one end, and similarly, you have the vector, and then you have the another end. So it will be ligated like this. So that's why we keep the temperature at 8 to 16 degree for ligation. Once the ligation is done, we keep overnight ligation. Next day we come, we take this mixture, we call this ligation mixture. We take this ligation mixture, okay. Now see what will happen to the ligation mixture. Initially what you have added, the insert and the vector. So you will have a position where you will have a vector. Okay, some molecules will have a vector and we will have a insert. Good, the thing that we want. In some cases, the vectors would have religated, right? And in some cases, you will have where the vectors are religated, your inserts will be freely moving, right? They'll be in the mixture, right? And now what you do, you take E. coli, DH5 alpha strain we usually use in our lab, okay? Yeah, BL21 if you want to express proteins, there are many strains. This E. coli, we have to first make competent, okay? We have to induce competence. Competence is that it should be able to take this DNA inside it. Okay, if they are not competent, they will not take the DNA inside it. In normal conditions, what happens, they have competence factors, they are proteins associated with the membrane of the E. coli. So this DNA binds with these competence factors, they have propensity towards them, and the competence factors then help the DNA to move inside the cell. This is the normal transformation that occurs in nature. But here, we have to force our DNA into the cell by forcing the cells to become competent. How we do that? We take our E. coli cells, okay, we grow them overnight, 16 hours at 37 degrees centigrade at constant shaking, like we did when we did plasmid isolation. But when we were isolating plasmid, at that time, that DH5 alpha had a plasmid inside it. This time, it's new. It does not have a plasmid inside. It's empty. It only has a genomic DNA. Okay. So what we do, we take E. coli cells, grow them overnight. Okay. Next day in the morning, we come and we put 1 ml of this E. coli culture into a fresh tube and keep it for 4 hours, constant shaking at 37 degrees centigrade. So in that 4 hours, what happens is your exponential phase is achieved. Okay. Competent cells are best made when they are made when the cells are in exponential phase. If you take the overnight culture for making competent cells, the cells will be in the stationary phase and about to die. Okay, they don't have good competence. So what we do, we take overnight culture, put 1 ml in a fresh tube, put it for 4 hours. In 4 hours, hopefully our exponential phase will be achieved, and we'll take these cells and put it in a 2 ml tube and centrifuge it. Okay, you have to handle them delicately. These competent cells are very delicate. So once you pellet them, what you do, you add 0.1 molar calcium chloride. Okay. Then you tap it and mix it. So these cells will be now dissolved into calcium chloride. Calcium chloride is the main chemical that helps in making competent cells. How it does, there's not a proof, but there are 2 hypotheses how calcium chloride increases the competence. So when we mix the cells, okay, and we have to keep this, do the whole process under laminar airflow, and this we have to, okay, do under sterile conditions, because contamination should not be there. So, and this all thing we have to do in ice. Okay, we take the cells, okay, centrifuge them. We have the pellet, throw the supernatant, that is a used medium. Then the pellet, we add 1 ml of 0.1 molar calcium chloride. Then mix it again, centrifuge it. This is called preliminary washing. Then your pellet will be here. Okay, throw the calcium chloride. Then again add say 500 microliters of calcium chloride, 0.1 molar, the same solution. Okay, and then resuspend the pellet. We take it to, we resuspend it gently. Okay, you don't have to be harsh, you don't vortex it. So gently resuspend it until you have your cells here. Okay, now you can keep it on ice for 5 minutes. Now you take this ligation mixture and add into your tube. So you take the ligation mixture, you add it into the tube, and then after addition, you keep it on ice for again say 5-10 minutes. Then what you do, some people keep it for 30 minutes. Okay, it depends upon how your lab does it. So once 30 minutes are over, you simply mix it and take it out, and then you put it at 42 degrees centigrade for 90 seconds. This is what we call as heat shock. We took the tube from ice that has our ligation mixture and component cells, put it in water bath for 90 seconds. It should not exceed 90 seconds. It should not exceed 90 seconds. So 90 seconds, you take it out and put back it on ice. Okay. Now you have a mixture. This is called as heat shock. It increases your transformation efficiency. Okay. Now you take this and add LB medium in it, say 500 microliters. Okay, and keep it at 37 for 2 hours. Okay, here what we're trying to we are trying to give this bacteria a good, nice, cozy medium, so that the heat shock and the cold shock that they had, they'll come out of it. Okay, they don't die, and the these host cells in which they have the plasmid, they'll be able to grow. So for 2 hours, we don't give any selection pressure. We simply put them in LB medium or a sock medium we can use. Okay, it's a highly nutritive media. We can use both. Put them here, and for 2 hours we keep it at 37 at constant shaking. After this, say you have added 500 microliters media. Okay, now you want to spread it on a LB ampicillin X gall plate, right? LB ampicillin X gall plate, you will pour this solution, but you'll not pour 500 microliters. There's a lot of solution to be added on a plate. So we usually use not more than 100 microliters. So what we do, we have to use all. So what we do is simply spin it. Spinning, what will happen? You'll have a pellet here, and then you'll have a supernatant. So we'll discard 400 microliters of this supernatant, and 100 microliters we'll keep here. And slowly, with the help of a pipette, we'll resuspend this pellet here back and take this 100 microliters and spread it on this plate. Okay. So now what will grow on this plate and what will not grow on this plate? So one condition, as I told you, you have a plasmid vector that will have an insert. Okay. That was transformed into an E. coli cell. Another case was your realigned, or what you call religated plasmid. Okay. It went into got transformed into the E. coli cell. Another case was your simply insert was there. Okay, it got transformed into the E. coli cell. Okay. Another case might occur where you have one vector, you have another vector. So you digested this vector with equar1, right? you digest this vector with equi-1, right? Now either an insert will go inside this or they will religate. But it can also happen that this is complementary to this. So they will be able to, because it has the same restriction site. So they will religate and they will religate, right? This will give us false positive results. Because in this case, your gene of interest will be not there, but the plasmid is there. You have a vector, you have a plasmid, again, another plasmid here. And it has changed the configuration of lag Z. Now lag Z is not functional, but there you'll be getting a white colony. Then you're confused what will happen, right? So these are false positives that you have to take care of. So this will be able to grow on this on this plate, and will have a white colored colony, right? These will grow, but they will have a blue colored colony because of your marker for screening or recombination. This will not be able to grow because it does not have an antibiotic resistance, plus it doesn't have an organism replication, it is not dispensable, so it will die. So these cells will not be grown. Now you have another cells in which you have a plasmid vector ligated to another plasmid vector, and now they are inside a cell and they will give you a white colony, right? So once this is done, next day you spread it on the plate. Next day you come, keep this plate at 37 degrees centigrade, okay? And then you have these colonies. You now pick up these colonies, your white colonies, and you again grow them in your 5 ml medium. You take tubes, okay, say LB tubes, okay, with kanamycin, sorry, with ampicillin. All will have antibiotics. Take this colony, the white colony, colony number 1, colony number 2, colony number 3, colony number 4. Okay, then grow them overnight. Next day you come and you isolate plasmid DNA isolation by alkaline lysis that we did the other day, right? And then you check for your inserts. How we do that? When we run these plasmids, how many bands we'll get?
3.
Multiple bands. Okay. Some plasmids give 2 bands, some give 4, some give 5. Your PEGFE that we used the other day gave 3. Okay, so we say always it gives multiple bands. This is the right answer. Okay, so you will have bands here. Okay, that means your transformation is successful. Now to check your cloning, what we do is, for example, this is your vector and this is your insert. Okay, this is your vector. Okay, this is one side of vector and this is your insert. Now initially we digested this with equi-1. Now my question to you is, can we be able to digest it again with equi-1? Can we be able to again digest it with equi-1?
Yes, sir.
Yes, we'll be able to digest the equi-1. So what we do, we take our plasmid, we digest it with the equi-1 again. Okay. Now how many sites of equi-1 are there? How many sites of recognition sequence of equi-1 are there? One is this side, on one side of the insert, and another one is on this side, other side of the insert. So you have 2 recognition sequences for your equi-1, right? Because we have ligated, the equi-1 sites have reconstructed again, because insert की भी sequence वही थी और vector की भी sequence वही थी. So when we digest it, equilibrium will digest here like this. So, and when we run the gel again, you will have how many bands? How many bands? 2 bands, because this has cut here. This will give 1 band that will be like this, the vector band, and this will give the band that will be the insert band. You will get 2 bands if your cloning is correct and if your insert is correct. And by this, and the molar ratio of the both will be same, that these to both bands should be of the same intensity. Okay, so that means you have cloned this insert in this vector. Now, for example, your vector was, you got a vector colony where the vector was religated. When you restriction digest it, you will only get 1 band that will be of the vector, but no insert. Right? Now you have a condition I just told you, you have 2 vectors, right? They have 2 restriction sites. This will give, this is a vector, this is a vector. So you have now 2 vectors. They have the same size because they are the same vector, because we started with the same vector. So you'll get 1 band here, and that will tell us this was a false positive. So cloning is confirmed when you restriction digest it, and you'll see that you have 2 bands. There's a so molar ratio. 1 band corresponds to the size of the vector. If you have a control vector, if you restriction digest it, you have a band here that will tell you that your vector band is correct. It should be at the same molecular weight position. And then the empty vector will not have an insert, but the recombinant vector will have an insert that will tell that your plasmid has been, your gene of interest has been cloned. Okay. Now, next question arises, how we do cloning of a gene. Okay, this was in case of eukaryotes. Sorry, in prokaryotes. Prokaryotes, cloning is easy because they do not have introns. However, in eukaryotes, you have introns. So that means a gene is larger than it has to be, because there are intervening sequences in between the gene. So what we do here, we do not, in case of eukaryotes, we do not clone the DNA, but we clone the mRNA, because your gene is this, it has introns, it gets converted into hnRNA, it has again introns, then these introns get spliced out, and then you have your messenger RNA that is smaller. So we clone this messenger RNA or the functional gene in case of eukaryotes, right? Or we can use genome DNA for cloning of genes that do not have introns. So that should be all about today's lecture about cloning. And now we're open for discussion, questions.